Yeah, now this is on vaccine basics, but this is in general just how do we develop immunity and based on immunity from viruses. This doesn't talk about the vaccines yet. This is just the general theory of acquired immunity. So the body's adaptive immune system can learn to recognize new invading pathogens such as the coronavirus SARS-CoV-2. So you have a coronavirus like this. Again, remember, it's a bundle of protein. That means it's a bundle of DNA or RNA, in this case RNA, that is enclosed inside protein and some lipids around it. So you have this so-called virus, or this bundle of genetic material, enters the body. Now you have a so-called spike protein on the surface and this M protein in between them. You have RNA over there. And there's there's the body. Now it enters the body. Coronavirus infection. The virus uses its surface spike protein to lock onto ACE2 receptors on the surface of human cells. Once inside these cells, these cells translate the virus's RNA to produce more virus. So human cell has these receptors here. This is called ACE2, and the spike connects to it. And just a star here is a note here. So the star means simplified. Yes, indeed. Anyways, so the virus goes into the cell through this ACE2 receptor. So virus enters the cell, and then now you have, then it creates a vesicle. So once it goes in, now you're going to have a barrier. Yeah, it's going to protrude it out. Now you're going to have a barrier inside of it, and you're going to be pretty much a vesicle with that membrane around it. And now it's enclosed like this in that vesicle. And then the genetic material gets released. So virus fuses with vesicle, and its RNA is released. So it fuses together with it as just a blob inside. And then virus fuses with vesicle, and its RNA is releases in there. And viral RNA is translated into proteins. Yeah, so somehow this gets over into the ribosomes. And then they translate it to proteins, and so virus assembly right here. Yeah, so it releases it goes over here. Now you have a new virus. It gets released. The virus released after it replicates itself. So immune response. Immune response here. Specialized antigen-presenting cells, or APCs, engulf the virus and display portions of it to activate T helper cells. And here's the diagram right here. So virus ingested by antigen-presenting cell, APC. So you have, so basically antigen-presenting one, and then it grabs onto this that's released because it's supposedly a foreign material. So it grabs onto it, and then it has portions of it sticking out there. So this displays portions of it to activate T helper cells. So there's a viral peptide that's presenting it over there. And then going next here is T helper cells enable other immune responses. B cells make antibodies that can block the virus from infecting cells, as well as mark the virus for destruction. Then you have cytotoxic T cells identify and destroy virus-infected cells. So what it ends up having is, on this antigen-presenting cell, it presents these peptides, and then you have the T helper cell connects to it, and now you have setups over here. You have the cytotoxic T cell, which can recognize this and go and kill infected cells, destroy infected cells, like that. Yeah, while the B cells here make antibodies that can block the virus from infecting cells. So this B cell, the B cell makes these antivirus. So once it gets this peptide over there, it knows it. So then antivirus, anti-coronavirus antibody. So it makes these antibodies that can connect onto the virus on the surface, and then these can't infect other cells. So it prevents virus from binding or tags it for destruction. So yes, it prevents it from binding now, or you tag it now, other ones can go and destroy other cells, etc. So, and then what you have is long-lived memory B and T cells that recognize the virus, can patrol the body for months or years, providing immunity. Simplify. So once they have these recognize these parts of that virus that's engulfed by this antigen-presenting cell, and then you'll have these long-lived ones, B and T cells that live long and have recognized it. So once it sees it, it goes in and neutralizes or creates antibodies to prevent infection and so on, and also destroy other cells that are so-called infected. And this is from nature.com. So yes, it's a very it's a pretty good illustration of just the mainstream theory of this so-called this germ theory, or not necessarily germ theory, just the idea of disease transmission. Now let's take a look at this figure illustrating the differences between all these. So this is from nps.org.au, an Australian government website, or just an Australian website right here. Yeah, nps.org.au. And let's take a look at this. So conventional approaches. Yeah, so these are conventional approaches to the SARS-CoV-2 virus and the vaccine production. So you have the conventional is protein subunits. So you're just using parts of the protein of these parts right here. So you would take some of this part here, and some of these other, maybe this part right here. So protein subparts that are, these proteins are connected to these. This RNA or DNA. It's in this case RNA. So you would get those over there, and then use these and inject it into the person. And those ones are subunits, and then you get an immune response where there are, the B and T cells can recognize these for future infection. And then you can also have this virus-like particles. You can have this set up here just with the spike, but nothing else, no genetic material inside, just that. And then you can also use whole virus vaccines. And this is an attenuated or weakened virus, and this is an inactivated virus. So this is attenuated, so you can make it weaker by removing this or switching this up so that it doesn't do the bad stuff, or it doesn't replicate, etc. Yeah, or this inactivated virus right here. Yeah, this inactivated could be set up like you could break it apart and etc. So it's pretty much the same thing, but the genetic materials modified. We'll get to this anyway. This one's weakened. This one I think will just be not replicating. Anyways, continue further. And now you will have is novel approaches. You have these two setups here. So you have the nucleic acid vaccines. So you have the DNA vaccine, is just the plasmid that you're throwing in there. Now you have the mRNA vaccine. You have that ball of fat with RNA inside. Then you inject it into the person. And this RNA is to code for specific antigens, such as these so-called spike proteins. And this one too could be coded for the spike protein, or this as well in the RNA side. Other one is the DNA side. Remember the genetic transfer where you're transferring via plasmids, etc. If you go all the way back to here, this one's an interesting illustration here. So you can have horizontal transfer. You get the plasmids transferring from different organisms to other types as well. And then you have mitochondria. You can also have virus transmission as well, as it illustrated here. Yeah, so in evolution, viruses are an important means of horizontal gene transfer, which increases genetic diversity in a way similar to sexual reproduction. So very, very interesting. So keep that in mind. Yeah, so when we're dealing with viruses, vaccines, especially DNA and RNA vaccines, you're dealing with genetic material transfer. Yeah, it's very interesting. And now the next one is a viral vector vaccine. So you have a non-replicating viral vector. You have something that looks like this, and so it's a different virus right here. Yeah, so it's a different virus that's containing the genetic material to encode for specifically this one here. And now this one, remember, so these ones are different colors just to illustrate that this is a different virus holding it. So this is a replicating viral vector that, you can replicate. This one doesn't replicate. And they both encode for that specific part of this one here. But they use different viruses there. So viral as the vector. So use different viruses as the vector. So this is from April 2020, just to keep that in mind, because it could be more updated types there. And now let's look at further on this protein-based one. This is the protein-based one. There's several types over here. These are conventional approaches, and the protein one is protein subunits. All right, so we have this. We could even zoom in. Let's just zoom in a bit. So protein-based vaccines. So you have protein subunits is one type. So you have 28 teams are working on vaccines with viral protein subunits. Most are focusing on the virus's spike protein or a key part of it called the receptor binding domain. Similar vaccines against the SARS virus protected monkeys against infection but haven't been tested in people. To work, these vaccines might require adjuvants, which are immune-stimulated molecules delivered alongside the vaccine. So that's interesting. So you have another immune-stimulating, but they're not part of the vaccine, or not part of the vaccine. But you're injecting it with the vaccine as well. I mean, they're not part of the so-called genetic material or the virus material that you want to develop the specific acquired immunity for. But this is just to stimulate the immune system. So as well as multiple doses. To work this, you might require adjuvants or as well as multiple doses. All right, so now we have this M protein, there's a spike protein, and just a note here, so most are focusing on the virus's spike protein or a key part of it called the receptor binding domain. So that one, I believe that one they're talking about this part right here. Now you have the spike protein on this end over there. It's interesting. Yeah, and here I just teleported higher up. So you see the spike protein on top there. There's an M protein at the bottom there. So it's very interesting. And continue to scroll back down. This one here has even more setup here, so it has extra stuff on top there, so you could use some of that as well. But anyways, we're going to go over here. So the spike protein, M protein, and then you put that in the vaccine, so you inject into the body, and then what happens is these particles, the antigen-presenting cell, or the APC, it finds these, and then now it binds parts of those onto here. So coronavirus peptide, and then there's an immune response, and they recognize, the T and B cells recognize this over here, and then develop immunity from that, or memory from it. And then you can also use virus-like particles, which is still part of the protein-based vaccines, because you can so empty virus shells mimic the coronavirus structure, but aren't infectious because they lack genetic material. So 5 teams are working on virus-like particles, VLP vaccines, which can trigger a strong immune response, but can be difficult to manufacture. And let's go to the bottom here. Actually, no, this is all, I think it's all from here. This is all from this figure. I believe it's from here. And this is on, COVID-19 vaccines and the research from April 2020. All right, continue further. All right, yeah, so this is, so 5 teams are working on this, or we're working on it, which can trigger a strong immune response, but can be difficult to manufacture. Because you got to manufacture, just an empty shell to mimic that virus. And that's over, it could be, right here, virus-like particles there. That's on that part. Continue further, and so you injected that in, this goes in, and then the peptide, coronavirus peptide goes over here, and there's an immune response. So notice there's always this setup over here with this antigen-presenting cell there. Very interesting. All right, so now let's continue even further, and now let's look at the virus vaccines. So we had, this was the protein one here, and now we're going to be the whole virus vaccines. This is the protein subunits, virus-like particles. Now you actually have the whole virus one, and you're using, this is the whole virus one actually. We have weakened one or inactivated one over there. So let's go back here. So virus vaccines. So weakened or attenuated. So a virus is conventionally weakened for a vaccine by being passed through animal or human cells until it picks up mutations that make it less able to cause disease. And this is Codagenics in Farmingdale, New York, is working with the Serum Institute of India, a vaccine manufacturer in Pune, to weaken SARS-CoV-2 by altering its genetic code so that the viral proteins are produced less efficiently. So it's weakened, so it passes through animals constantly until it weakens via mutation. So very, very interesting. So and then you put that in the vaccine, and you can do the same setup here. But then here's inactivated virus. So in these vaccines, the virus is rendered uninfectious using chemicals such as formaldehyde or heat, making them, however, require starting with large quantities of infectious virus. You got to get a whole bunch of them, and then you're going to weaken them, etc., with heat or chemicals, and so on. And then the same thing, you insert it into the cell, into the body, and then you have these antigen-presenting cells go in and absorb that, engulf it, and then it gets the peptides or parts of it and presents it for the immune response system. So there's coronavirus peptide, and so on. So the setup is always the same there. So now let's look at viral vector vaccines, and that's over here in the diagram is viral vector on this setup over here. And then the next one is nucleic acid vaccines after that. This is viral vector one, and these are the novel one, new types. And this one uses other virus as vectors for coding for that specific virus you want. Let's go back here. All right, yeah. So viral vector vaccines. Remember, you're using other viruses as vectors to deliver genetic material of the target virus. So replicating viral vectors such as a weakened measles virus, you use that as the vector. So the newly approved Ebola vaccine is an example of a viral vector vaccine that replicates within cells. So such vaccines tend to be safe and provoke a strong immune response. However, existing immunity to the vector, not the target virus, could blunt the vaccine's effectiveness. However, that's interesting. So you'll have a weakened measles virus like here, and there's a viral gene, etc. What you're going to do is you modify it so that the spike that it produces is actually the coronavirus spike, and this one can replicate and so on. So it's the target one that you want to build an immune system for the spike ones to recognize the coronavirus. But however, this one, if people already have immune response to the viral vector, then this might get destroyed quickly, and this won't be as effective of getting this into the immune system. And let's just erase all this. And now let's look at the non-replicating one over here. So non-replicating viral vector, such as adenovirus. So there's currently no licensed vaccines use this method. So yeah, you'll use this adenovirus as a viral vector and it's non-replicating. But they have a long history in gene therapy. So those are modifying the genes if you want to treat for cancer, etc. Booster shots can be needed to induce long-lasting immunity. That was just pretty interesting. So booster shots, subscription-based vaccine induction. So U.S.-based drug giant Johnson & Johnson is working on this approach. So what you'll have is a coronavirus spike over here. Again, you want to code for that. There's viral genes somewhere inactive, so to prevent replication. And then you'll have the vector right here, such as adenovirus. And this one here is weakened measles. Yes, and in both of these cases, what you're going to have is when you inject it in, and again, you have that, so virus replicates in this case, but in this one, it wouldn't replicate. And then what you'll have is, the antigen presenting cell engulfs these, and then it puts the coronavirus peptides, or in this case, the spike peptides, a specific one that you want in to be recognized. So it'll put some of those antigens, some of those peptides on here, and then you get the immune response and so on. So that presents it to the T and B cells. They go and look at it and memorize it for a future. All right, so now let's look at the nucleic acid vaccine. So now you have the two types of setups right here. So you have the DNA vaccine and the RNA vaccine. And this is why I went over the electroporation, just to make it easier for the cell to go into, for the vaccine to go into the cell. So what you're going to have is, you're going to have a coronavirus spike gene. This one's just a plasmid with that specific spike gene that you want to be recognized. And you put that inside over here. So that's a DNA plasmid, and there's the electroporation there. So you're going to put this in a vaccine. And then what you're going to do in this case, you're going to insert it in, and usually including a process called electroporation, which creates pores in membranes to increase uptake of DNA into cell. Because you're basically putting a naked plasmid inside the body, and then you want to weaken the cells around using just an electric field to break it apart, so that this can go all the way inside. Now you have a plasmid going to the nucleus, and then what happens is once it gets into the nucleus, it will translate into mRNA, or transcribe into mRNA, and then this goes in ribosomes and translates into that specific spike protein setup over there, and then this goes into that antigen presenting cell for the immune response. So again, that's the same cycle over there. All right, so now the next setup is RNA vaccine. So basically, you're going to have a ball of fat with the RNA inside. So you have RNA is enclosed in a lipid coat so it can enter cells. And then there's the RNA with the specific spike protein gene over there. And then what you can do is throw it in there, or spike protein sequence that's transcribed from the DNA already. And so you're going to put that in there. And then you're going to inject it, and this goes in because it has that lipid layer. It can go in here, connects to the cell, and then they can deliver this inside. And then this goes to the ribosome and translates it into proteins. so viral proteins, etc. goes over to here, and there's a coronavirus spike peptide on the antigen-presenting cell that engulfs these. And then again, you get an immune response. Again, this is from April 2020. So if RNA and DNA-based vaccines are safe and easy to develop, to produce them involves making genetic material only, not the virus. But they are unproven, no licensed vaccines use this technology. This is from April 2020. But the RNA one does, and I believe the DNA one has been. Yeah, the DNA also has been approved in India, and the RNA is approved in the US and so on. And the keyword here is it's easy to develop. So right now this is from April 2020. So this is unproven, no licensed vaccines, but they are licensed now in some parts of the world. So the keyword is easy to develop. So expect a whole bunch of these vaccines and booster shots, etc., to be on the way. Because if it's easy to develop, that's going to mean more sales. For big vaccine manufacturers.